Okay, good morning, everybody. Once again, thanks for all of you for joining us, as usual. We're just, as usual, going to give a minute or so just to let all of you join us. As we've been doing, we will continue to just do some updates. This week, we have some brief updates. And of course, as every week progresses, we will provide updates as needed from all the panelists that are with us. We still have a large number of panelists who are on the webinar side to answer questions. So we'll once again be answering your questions throughout the webinar. Any question you might have, just put them in there. And once again, we'll take all the questions within a day or so. We'll get them sent out to you via email, and they'll be posted on our website, the Department of Medicine website, for you to come back to. For those of you who may not be getting the email, so some of you joined via the COVID-19 School of Medicine email. If you aren't getting the follow-up emails from our grand rounds, feel free to shoot me a message, and I'm happy to add you to our email distribution lists, and you'll get updates from us directly. And so we'll have the follow-up with the video as well as bullet points. Thank you, Joy, for making those every week, and send that out to you. Just FYI, for upcoming grand rounds, we have a lot of great topics coming up. We will be talking, we've been getting a lot of questions in grand rounds about some of the effects we're seeing and learning more about in children, questions about OB-related complications. So that'll be the topic for next week. The week after that, Dr. Manisha Desai-Will Collins will be looking at, who've been looking at the trends of the characteristics of our COVID-19 patients at Stanford. They're going to be talking more about that. So we'll get to learn more about what are the patients that we've been getting at Stanford with COVID-19, what are the characteristics of them, and what can we learn about of our patients, essentially. And then the following week, we'll have a great lecture about vaccines led by Kari Nadeau and actually Scott, who's with us, and a couple other faculty. So I'll talk more about that in the coming weeks. But the goal there is to help us understand vaccines, what are the obstacles, and what's it going to take for us to get to that vaccine that, of course, we've all been waiting for. So with that being said, I'm going to turn over to updates this week. We'll start with Dr. Ann Weinacker, and Scott Boyd will provide us updates. But Ann, I'll turn it over to you.
Great, thanks, Errol. Brief update today. There are 5 patients with COVID in the hospital, 1 in the ICU on a ventilator, and there's 1 patient who is a PUI. The good news is that overall, particularly in Santa Clara and San Mateo County, the number of cases is going down. And in the process, or in the meantime, we are ramping up in terms of the number of elective cases that we're doing. Last week, there were nearly 1,000 cases done, elective cases done. And this week, we're on track to do about the same number of cases. The clinics have been gradually ramping up, sort of unofficially, but there will be some recommendations made formally to court tomorrow from the ambulatory transition team regarding opening sort of officially on Monday back to in-person visits in the clinics. And it's really, the recommendations that they have are really around making the environment safe, so cleaning and masking and those kinds of things. The clinic managers and the clinic chiefs are working closely together to make sure that the environment is safe, both for patients and for healthcare workers. And then the last thing I wanted to mention was the visitation guidelines have been loosened a little by the county and by the state, such that now they are suggesting, and we are going along with, of course, allowing patients who have, adult patients who have physical, intellectual, developmental disabilities or cognitive impairments, to have a support person with them, and up to 2 different people can be involved in visitation, but no more than 1 person at the bedside at a time, and nobody can spend the night. Beyond that, I don't have anything new to say.
Thanks so much, Ann. Scott?
Yeah, thank you. So I wanted to just give a brief update on things that we've been working on in the clinical lab to help with the serology analysis and understanding the patients that we have at Stanford. So I'll show this. Can you see my slides?
Yep.
Okay, so yeah, this is the laboratory colleagues who are mostly involved in this. So as you know, we've been presenting data up to now from a regular ELISA that we implemented in the lab. We've been measuring IgG and IgM. We've also been testing and validating some commercial assays as they become available. There's still questions about the supply of those, but that's something that we'll stay on top of as we go forward. I wanted to show everyone, these are some data from actual Stanford patients, so Stanford inpatients, just so everyone can get a better sense of what the serology looks like in people who are ill enough to present for hospital care. There are a few outpatients in this mixture, but many of them are admitted or ICU individuals. And so this shows days since symptom onset for the IgM, IgG, and IgA, which we've been doing in a research context in parallel for about 62 people with about 320 time points sampled overall amongst the whole group. And so I think some of the interesting things that we've seen already in the relatively early days of this pandemic are that the time at which the antibodies appear for most people, again, in this relatively sicker set of individuals, is not that different for IgM, for IgG, and for IgA overall. So there's not like a long lead-in period with IgM only in most individuals that we see. And something that's struck us is that the sort of appearance of IgA seems to be a bit more variable between different people. And even within the first month after the onset of symptoms, we see that the IgM levels look like they're starting to drop, probably the same with IgA, but the IgG continues at a relatively high level in these people that we've observed. And an important aspect about these kind of measurements that we've been making and reporting to everyone here is that they're measuring just the antibody quantity, but not the quality or the nature of the exact antibodies that are present in the mix. So something that we've been working on now as part of a great collaboration with Peter Kim's lab and Abby Powell in that lab in particular, Katrina working with me, is sort of a continuation of the great collaboration we've had with the ELISA development that involved many Stanford faculty that we've talked about in the past presentations. So what we've been testing is an assay that would see whether the patient's antibodies can block the interaction between the viral spike receptor binding domain protein, which is the antigen we've used in the ELISA, and the ACE2 protein, which is the host cell receptor as a purified protein in this case. So in general, if you have RBD protein on a plate and you add the ACE2 protein, it binds nicely. If you add patient serum to the well before that, you get two kinds of results or something in between. So if someone doesn't have blocking antibodies that prevent this interaction, as in this case, then the RBD binds just as before, and then we can detect it with a secondary antibody that will bind to, in this case, an FC fusion that allows us to pick it up. If a patient has a high levels of blocking antibodies, in that case, when we add the RBD, it doesn't bind, and so we don't detect any signal. So we've been testing that out recently, and I think the results look promising. I mean, as yet, we don't know whether this is a great correlate of protection, but it's actually probably more informative than the total levels that we've been looking at so far. So this is data from one patient. You can see the increase with the blue being IgG, red IgA, yellow being IgM, as the person increases their days since symptom onset. So in this case, the three come up relatively similar timeframes. That's something that sort of levels out after a while. This is the measurement performed by Ben Pinsky and the virology lab folks of the viremia in the blood of this person. So it starts off having CT levels that are easily in the detectable range, but then as the antibodies come, the viral RNA drops. And so there's a removal of the RNA-emia in this patient. And here are the results of that blocking assay. So this shows the ACE2 binding at each time point along the way, synchronized with this. So really, once the antibodies start to appear, then we see this ability of them to block the ACE2 binding and going down to pretty near complete suppression of that interaction. So that's something that we're hoping to be able to implement as a potential clinical lab assay. It's sort of a new element that we should be able to bring to the whole interpretation of the sera of the patients. We've tested a limited number of outpatients so far at relatively early time points, and so far we haven't seen a lot of blocking activity, but maybe there's a trend for it to be showing up at lower levels as we go further out. This is far from statistically significant or representative, so that's something we'll be working on as we go forward. Then, so finally, we've also been asked just in general to try to think of ways to scale up our serological assay capacity, just as the PCR capacity is being asked to increase a lot, and have been given the goal of being able to do 20,000 tests a day. So that's a big stretch from where we are now, but we're working on ways of doing that, and with a lot of help from the hospital and the whole community. We think that probably for the purpose of seeing whether people may have immunity testing IgG only would be a way to both increase the scale and focus on the antibodies that are likely to be most important. And we're, as I mentioned, evaluating some commercial kits at present, which would also help us to increase our capacity as well as the lab developed test. And maybe that pooling strategies might actually help if we have low incidence or low prevalence populations to test. So we'll continue to work on this blocking assay, and in parallel, I'm working with Ben and others to try to see if we can have a cellular viral neutralization assay to complement this as well. So that's all I wanted to show today, and I'll hand it back to the Errol.
Thanks so much. I really appreciate you joining us every week with these really helpful updates. You and Ben will continue to stick with us, and we really appreciate updates every week. And of course, as I mentioned earlier, you'll be one of the main presenters a few weeks from now, talking about this more. So again, thanks so much. So with that being said, we'll move on to our main presentation today. A quick update, we won't have any CME this week as per our main presenter is coming from Biotech, but we're more than happy to make that concession having Phil join us today. Dr. Phil Pang is a physician. He actually is an alumni at Stanford as well. He did his BS here many years ago, and then went to Columbia both for a PhD and an MD in biochemistry and biophysics. He has been big part of biotech for many years, including at Gilead as one of the leads there, and eventually went on to be a CMO, Chief Medical Officer, at different companies, including now at Vera Biosciences. When I heard about Dr. Pang, learning about what he's done, and mentioned his name to anybody who knows him, people were, no pressure, Phil, people were really excited to hear you come and speak today. Appropriately so, ever since COVID-19 came, we've been relying heavily on biotech in many forms, and so we felt it was very appropriate to have you come and talk from your perspective, a biotech perspective, about the approach to COVID-19. So thank you so much for being with us, and looking forward to hearing what you have to say.
Thank you so much, Errol. And I just want to begin by thanking everyone in the Stanford community for this invitation. It is a little bit unusual, I'm sure, to have someone from industry speaking in this type of forum. And actually, just to highlight, I'm not just an alumnus of the undergraduate program, but I actually did my internship, residency, fellowship, and postdoc at Stanford as well. And so I actually wanted to take this opportunity to say it's an honor to be here amidst so many of my former mentors and teachers. So thank you very much for this opportunity. So, of course, I am from industry, and so I want to begin by the fact that we are a publicly traded company, and so therefore I have to make the basic statement that when I say may, it means may, and doesn't mean that it will happen, which is, of course, sort of a comical thing that we have to say in industry. I also do want to state a few disclosures. I am obviously an employee and shareholder of Vera Biotechnology. This is an endeavor, especially with regard to COVID-19, where we have many collaborators, and I'll talk a little bit more about why that we believe that that is so important. And of course, one of them is GSK, and GSK wanted me to highlight the fact that these are the opinions of Vera and myself and not GSK. So moving on from that, I wanted to begin with the questions that really come to my mind and provide sort of a very what I consider to be a pragmatic view of what the future looks like and what we need to be keeping in mind. And that is, where are we going? When will we get there? Who will get there? And what role will antibodies play? Of course, Vera is an antibody-focused company, so that is something we think about a lot. So in terms of our destination, I really like to bucket this into 4 major destinations, or what I would call indication populations, which is, of course, prevalence and vaccines, which were all top of mind for us, but also 2 other indications for peri-exposure prophylaxis. One can imagine, as you know, that many patients are dying in our nursing homes. These are the elderly, in whom vaccines may be less effective and in whom immediate protection would be obviously invaluable. There's also the question of, can we take these patients who test positive or who have mild symptoms and prevent them from progressing into the hospital? And finally, on the right-hand side, obviously those who are already hospitalized and have severe disease. 2 things of note: the population, obviously, the number of patients in each one of these buckets diminishes. Obviously, healthy volunteers on the left, hospitalized patients on the right. So the impact we can make, it varies depending on the population size as well as the medications available. And on the right and on the top, I also want to highlight the clinical challenge. I think as physicians, we are all well aware that early treatment or even prevention tends to be a much more profound way to move the needle, whether or not it's oncology, getting to the tumor early, whether or not it's cardiology and trying to intervene before there is sufficient vascular disease. So I think that holds true for infectious disease. And so this biological or clinical challenge is intersecting with the populations at risk and where the need is. And that's why I like to say this pandemic will require many success stories. So the good news is that even if this pandemic requires many success stories, there are many possible success stories. So the good news is that right now, per our calculations as of 2 days ago or 3 days ago, there are over 350 medications, or I should not call it medications, drug candidates and or medications being pursued in the COVID-19 space. I think to summarize, more than 90 of them are repurposed medications, more than 90 vaccines in development. That's of course, as of, as I said a couple of days ago, that number is increasing literally on a daily basis. And there are nearly 160 fit for purpose COVID-19 therapeutics that are either preclinical or planned to be in the clinic very soon. That's the good news. The bad news is, of course, once you have over 350 medications being developed simultaneously in a very short time frame, this leads to crowding. The FDA has indicated to us they are receiving more IND applications in the last couple of months than they usually receive in an entire year. Sites will then start to feel this type of pressure, how to choose between these many promising medications or promising drug candidates. And of course, that can lead to bottlenecks at IRBs, et cetera, et cetera. So I think of this as a great opportunity because there are so many success stories that we will need. But at the same time, this is some of the pragmatic challenges that we are seeing ahead for companies. And I do want to talk a little bit more about that. So when will we get there? So I want to highlight a few things on this slide, and actually realizing it's just a build, give me 1 second. I think that obviously repurposed medications will get there first. We see this with, technically remdesivir is not a repurposed medication since it was never approved, but as you know, it was tested in the Congo for Ebola, and there was stock available to start testing right away. Hydroxychloroquine has obviously received a lot of news. There's obviously studies for convalescent plasma. There was the recent Regeneron study with IL-6, and these will make it into clinical practice and potentially eventually achieve a label indication, and these will be available now or in the near-term future. I think the other two major areas that I will discuss further today is antibodies, and the reason for that is, for example, there's been a lot of companies, including ours, but including Regeneron, Eli Lilly, AstraZeneca, and so on, who have really accelerated the development of monoclonal antibodies. Many of these will enter testing this summer, and it may be possible, therefore, that they will have an emergency use authorization by the end of this year. And I'll talk a little bit more about that. Now, these later boxes of when conditional approval will happen or actual antibody approval will happen are really speculative. This will really depend mostly on the regulators as to what they will consider acceptable for an actual license as opposed to an emergency use authorization. And that's true also for vaccines. We've talked a lot about the fact in the press that Moderna and now I think the other vaccine that's gotten a lot of press is the Jenner vaccine from Oxford, now licensed by AstraZeneca. That will either have started or will start soon, all those trials. And we hope to have readouts from them by the end of this year. At least that's what these companies are saying, which could lead to obviously an emergency use authorization and vaccine approval, depending, again, could be as late as the second half of next year or earlier, depending on when the regulators see fit to and what type of package they're looking for. Now, amidst all of this, I'm going to sort of double click first a little bit deeper on vaccines and then go into antibodies, but purely from a more of a pragmatic perspective rather than a comparative perspective. So onto prevention. Obviously, we all know the axiom, an ounce of prevention is worth a pound of cure. I do want to highlight, though, that in addition to being safe and low cost, it needs to be available on the scale of billions of doses. And that is a challenge that we have rarely faced as a society, let alone for any one company. So I do hope that there are many success stories because the ability to achieve a scale of billions of doses is is an incredible reach. And the sad reality that we are seeing is that even if a company were to be able to reach 1 billion doses, which frankly, the only company so far who believes they can get there is J&J or Janssen, 1 billion doses will still leave 7 billion people without access to this particular vaccine or that particular vaccine. So I think that we are really in a challenging time here, that even if we were able to achieve the kind of scale we think is even potentially possible, we may leave many people behind. So with that, I want to talk about some of the challenges as I see it from a vaccine perspective. I am not a vaccinologist, so these are very high-level pragmatic considerations. On the right, what you see is the fact that there are many, and this is already outdated because it's 2 weeks old, many different types of vaccines that are being put forward. And I'm not even going to go and attempt to say which biology is better than what biology, because frankly, I don't think we know. And we'll just have to find out empirically. There are, of course, theories as to why certain vaccines might work better than others. But I actually want to take a moment to look at the left-hand side of this slide, which is to say, there are some basic things I think of from a practical perspective of what we need to think about when we think about a vaccine that's going to be successful. And that is, what does immunogenicity even mean? We heard earlier today from Dr. Scott Boyd around serology testing and what that means. It's a very similar issue. If we give a vaccine and we're hoping to mimic the infection or the serology achieved by natural infection, what does that serology need to look like? Second, from a company's perspective, there are many companies who can do a phase 1 trial that is, let's say, 20 patients and look for quote unquote immunogenicity. But the real question is can they translate that immunogenicity into actual clinical endpoints? Those trials, I believe, will require tens of thousands of patients, and many companies are not simply able to execute on that type of design. Finally, we talk about crossing the finish line, and I'll just share with you a couple of quotes from some leading vaccine manufacturers. So the first is immunogenicity. So we saw some very interesting data already from Dr. Scott Boyd this morning around the fact that there is the quantity, but also the quality or the type of the antibody response that matters. And he showed you one example of using a blocking ACE2, using a blocker to the exact epitope to which ACE2 binds. That is obviously one of at least what we've cataloged as 7 different epitopes on the receptor binding domain, of which only one is the actual interaction with ACE2. So the question is when we look at the receptor binding domain, as illustrated on the right, we're seeing at least 7 different epitopes. And what you're seeing on the right is an epitope map where you take all these different antibodies, which bind on the far right-hand side of the screen, showing the Roman numerals, to different epitopes on the receptor binding domain. And if I just point you, I'm not sure if you can see, but if you look at the very top right-hand corner, and I'm going to try and get my pointer to work, for where, for example, S309 and S227 interact, you can see that there's an increase and then there's a second bump up. That means that the first antibody does not compete with the second antibody, and the second antibody is binding to a unique epitope on the receptor binding domain. This is early mapping data. This is now almost a month old. And what it suggests is that, first off, there can be neutralizing antibodies that do not block ACE2. Second, there are antibodies that bind outside the ACE2 binding domain that are not neutralizing. And also what we're really finding that is very concerning to us is that a lot of the pseudovirus neutralization assays are not correlating with live virus. And so actually what we've found is that yes, pseudovirus neutralization, which is a way of rapidly testing and easily testing in a non-BSL-3 facility, whether or not a certain antibody or serotype might block infection, the reality is unless you do it on live virus in a BSL-3, you won't know for sure. That's one of the reasons why, for example, and then there's another aspect to that, which live virus stock you're talking about. We are now looking at live virus stock on 3 continents, one in Asia, one in the UK, and one in the US, just to make sure that the serologic conclusions and or the antibody conclusions we come to are valid across these different regions. And finally, I want to point out that some of these antibodies bind, some of these antibodies neutralize, but may not have effector function. And we have still not described the role of the rest of the immune system, such as NK cells or macrophages pursuing ADCC or ADCP, which may impact the actual efficacy of any naturally occurring antibodies or any antibodies that we develop, not we, but the industry as a whole develop. So next, I want to move to a second pragmatic challenge, which is how do you power a vaccine study when the incidence is unknown? And I want to apologize to the statisticians in the room who may be cringing right now, but this is sort of a back of the envelope discussion where I want to say, you know, there was a recent study out of Korea suggesting that the incidence of symptomatic disease of a secondary contact, so I have COVID, I go have dinner, hopefully, I shouldn't have dinner, but I have dinner with a colleague, what is the likelihood that they're going to get symptomatic disease? And it was as low as 0.55%. The incidence among family members of COVID contacts have been estimated between 0 and 16%, which of course is almost a nonsensical range. The reason for that is of course, early on in the pandemic, there was not isolation within isolation. What I mean by that is that when a patient self-isolated at home, they did not isolate themselves from their family members. But now, of course, they're both self-isolating from the community and from their family members, and that has reduced the incidence in some studies, small numbers, to as low as 0% versus 16% when they do not isolate from family members. That's in what we can trace and contact from people who have a known index case. But what is going to be the general attack rate in the community? Will the vaccine prevent actual sterilizing immunity, meaning they will be PCR negative, or will it just prevent symptomatic disease? Of course, these numbers above are for symptomatic disease, but the incidence for PCR negativity will be higher, which will make the study easier to power, but also is a greater biological challenge. Social distancing. It's changing every day. It's changing on a country by country, state by state, on a county by county basis. How that will impact attack rates is hard to say. So really from a clinical trialist perspective, we need to be in the right countries at the right sites at the right time. And that is going to be a challenge in and of itself. We can tell you, for example, from flu studies, that when the CDC says it was a bad flu year at, let's say, 4%, some of the studies are coming in the placebo arm with an attack rate of 1%, and some with 10% even in the same year, which really emphasizes the need to be in the right place at the right time. So just to give people an orientation to what I mean about this problem, is as if we assume an aggressive attack rate of 1%, which I think is enormously high, and a 50% reduction in that rate to 0.5%, you would still need a 10,000 patient healthy volunteer study, 5,000 on placebo, 5,000 on the vaccine. So if you use that back of the envelope calculation, as you know, the Jenner Oxford AstraZeneca study announced 8,000 people. So basically they must be assuming a higher attack rate. And that is one of the risks that is being taken by that particular study. I think it's a great vaccine candidate, but of course, everything comes down to, can you execute on the clinical trial? To that end, I would also like to point out that most other companies probably will not be willing to take that risk. If they're going to bring a vaccine forward, they will probably shoot more on the order of 30 to 40,000 people in the vaccine study. And that, of course, is a challenge that only a few companies will be able to undertake. So then finally, I want to talk, about the end, which is, imagine you're able to have the vaccine with the right immunogenicity. You're able to show that that immunogenicity in a clinical trial of 30,000 patients is actually able to result in clinically meaningful endpoints. Then the question is, can you distribute and can you actually achieve the billions of doses necessary? And I sort of want to just give you two sort of tongue-in-cheek quotes that are sort of the diplomatic way to put it, which is, there's almost no capacity for most of the vaccine companies to actually do anything meaningful at scale. I don't think that that is meant to be anything other than just a recognition of the reality. I think there was this great panel I was on with Johan from Janssen, who said that everyone realizes that the bottleneck will be the filling lines. What that means is, of course, now you have the vaccine, maybe you've created 1 billion doses. Well, how do you actually bottle those billions of doses and actually distribute them around the world to people who need them? How many doses are you going to put per vial? Are there even enough glass vials around? These sort of things become very pragmatic considerations. And actually companies are spending billions of dollars right now trying to soak up and secure as much filling space as possible to be able to actually execute in the hope that their vaccine actually will work. So finally, I want to take the last 10 minutes to talk about monoclonal antibodies. And I want to talk about them from two perspectives, although I think that they can be used in both peri-exposure prophylaxis, mild to outpatient treatment, as well as severe hospitalized treatment. I want to focus actually on these middle two buckets just in the interest of time. And I think that the things I've highlighted is, although obviously a vaccine is what we are looking for to end this pandemic, as I've mentioned before, I think there will be many success stories and many indications that are necessary. And I want to highlight the fact that, as I mentioned before, vaccines may not be, I shouldn't say ineffective, I should say less effective in all patients. So, and there's always the risk, of course, as we've seen, for example, with hepatitis B vaccines, that the elderly may respond less well. We also see that with a number of other vaccines. Well, the elderly are those who are most at risk at this point. Similarly, for those large number of patients who do test positive, what can we do for them and to keep our hospitals clear and free of these types of patients and have them obviously accrue the benefits of not being hospitalized. So in that setting, one second. I think that this is data that is published from the MMWR, which I think everyone is familiar with, which is simply the fact that the elderly represent a third of the cases, nearly half of the hospitalizations and ICU admissions, and 80% of the deaths. I think everyone can quibble with the numbers at the right, the case fatality rates, and I know that they obviously are highly dependent on the denominator, which we still do not know. So I'm not trying to put this forward as absolute numbers, but simply a trend that the case fatality rate is clearly increasing with age. And I think there was recent public press around 1/3 of deaths in the United States occurring in nursing home facilities. So I think that there is a huge lever we can pull in that particular area. And that's one of the areas that Vir is certainly interested in, as well as many of the other antibody companies. So to that end, why monoclonal antibodies? I really like this slide mostly because of the picture at the top right. The top right is Davide Corte. You may know him from his work with Antonio Lanzavecchia. Davide is one of the leading B-cell antibody immunologists and virologists. And back in 2016, he isolated this antibody called MAB114. MAB114 was subsequently shown in that recent New England Journal paper to reduce the rate of Ebola deaths from, I believe it was 50 or 55% in the remdesivir arm down to 30 or some odd percent in the MAB114 arm. So I think, that is a maybe a salient example of the role that monoclonal antibodies can play. I think we're all aware of the role that antibodies in general can play in infectious disease. And really, one of the ways in which I like to explain it is it really is a 21st century version of convalescent plasma. Obviously, we're all aware of things like cytogam and these sort of things, but this is sort of the next generation version of it. So moving on from there, I wanted to address a couple of concerns about monoclonal antibodies and things we need to get in place. But before I do so, I thought this quote from Scott Gottlieb was a very seminal quote, which says that because of the timing and the rapid availability, the fact that the studies can be somewhat smaller, there's this belief that antibodies can be available in the summer, and I believe that that is now proven to be, are on track to be true, and that this, because of the populations it can go into, could really make an impact both in peri-exposure prophylaxis, early treatment, and late treatment. Doing so, of course, requires, again, manufacturing. And so that's the theme of the day, at least in my mind. This is an incredible manufacturing challenge, whether or not it's for remdesivir, which is a small molecule, but a complex process, or whether it's for monoclonal antibodies. For monoclonal antibodies, as you know, the time from what we call sequence to IND, which is the sequence of your monoclonal antibody, you send it in an email file to a manufacturer, or if you own your own manufacturing plant, send it to your manufacturing plant. How long does it take to then result in an IND? That traditional timeline is actually aggressively stated here by Brian Kelly at 10 to 12 months. Many companies often find it is closer to 14 to 16 months. And in this opinion piece, Brian has basically said, we believe we can reduce it to 5 to 6 months, not by compromising safety, but basically by spending more, by doing more things at risk and recognizing that that risk may play out in your favor or against you. The timelines recently expressed by both Vir this summer, Regeneron this summer, Lilly this summer, suggests that since obviously the epidemic is only 6 months old, these are clearly timelines that we believe we will achieve. To that end, there's also scale. So this talks about what I call fast to clinic. This slide more talks about once you achieve the clinic, can you actually manufacture it at scale? Again, the same problem for vaccines, trying to achieve billions of doses. Now, can you achieve millions of doses of an antibody at scale? I know that many people traditionally think of antibodies as being very expensive medications, but in the last 20 years, the yield of antibody manufacturing has increased dramatically on a log scale. And it is definitely possible to create millions of doses at scale at a cost that would be affordable and not astronomical. So we're talking not tens of thousands of dollars, obviously, which would be ridiculous in this type of situation. So the question then becomes, where do you get it manufactured? Many large pharma companies have their own manufacturing plants. And then there are what we call contract manufacturing organizations, which can manufacture for companies who do not have the entire capacity they need. And frankly, that's every single company will not have the capacity they need, because again, even if we're able to create 1 million doses, there's 1.4 million people in nursing homes right now just in the United States. So now you need to create 5 to 10 million doses just to cover the nursing home populations around the world, and then so on and so forth. So this is going to again be a place where even when we have the medications, there will probably not be enough for everyone. And that is something we as a society will have to continue to wrestle with. Finally, I want to conclude with some thoughts on, and I hope you'll indulge me here, where I'll talk a little bit about Vir's actual monoclonal antibodies. And I really like this quote, I rob banks because that's where the money is, as opposed to looking under the street lamp. And we've taken a somewhat different approach to looking at monoclonal antibodies in this space. First, I want to talk about the right-hand side, which is where you would imagine we should go and where most companies are going and where we also are going. We have over 150 blood samples from COVID-19 patients and all their B cells. We are looking at hundreds of thousands of B cells from those 150 patients and are isolating antibodies that are potent against COVID-19. But this is what we would call our backup or secondary approach on the right-hand side. Our initial approach and the antibodies we plan to bring to the clinic are actually from the left-hand side, which is to actually start with SARS-1 patients. And I realize this may sound somewhat counterintuitive, but there's a number of advantages that I'll talk to you about, which is that if you isolate a neutralizing SARS-1, meaning the outbreak that happened in 2004 antibodies, and they're neutralizing, and then you're able to demonstrate there are potent neutralizers of SARS-2 or COVID-19 or SARS-CoV-2, depending on whether you're talking about the virus or the illness, then you will have achieved two things, which is not only will you have achieved potency, but I think very importantly, you will have covered the evolutionary distance space between SARS and COVID-19, and therefore hopefully have a higher theoretical barrier to resistance, and therefore avoid the need for many cocktails of antibodies. To that end, that's exactly where we arrived at. On the left-hand side is a pseudovirus neutralization. Now, I've already told you that those are not a perfect correlate of live virus neutralization, but the reason why I show this figure is that it shows an evolutionary span that goes from in the dark maroon, SARS-1, to the light red, which is COVID-19. It also includes some bat sarbecoviruses and et cetera, et cetera. And for S309, we have basically, which is the antibody we've chosen, which you show potent neutralization down to around 100 nanograms per ml. Of course, it's important to show that it neutralizes the live virus, and that's what's shown on the right. Again, I draw your attention to the red line, S309, showing a EC50 around 100 nanograms per ml. One of the questions people have is well, is the live virus, is the virus mutating, and how does that play out over time? I want to say that, as I mentioned earlier, we are looking at live virus from 3 continents, both from Asia, the US, and the UK, and this experiment has been performed on all 3 sets of live virus. So I think from maybe delving into the science side of things, I think that this data suggests a couple of things about what we can predict from a structural perspective. That first, the epitope should be mostly invariant between SARS-1 and COVID-2 and COVID-19, given the fact that it neutralizes both. But it will probably not be where the ACE2 binds, since that this region is not conserved very well between SARS-1 and COVID-2. It should also suggest that when we do an analysis of the 1,200 available sequences of COVID-19, that this region should be highly conserved. And I wanted to show you some what I think interesting photos regarding that. So here is the epitope itself. And I apologize, I'm missing a little a legend here. The blue is where ACE2 binds. This is the footprint for S309, which is the antibody we are taking into the clinic. And I want to show you some slides that suggest what is happening in this region. So first, if you compare COVID-19 to SARS-1, you can see that the region in blue, which was the ACE2 binding domain, is very different between these two viruses, but is not very different in the S309 footprint. In fact, there are only 5 amino acids that differ in this 21 amino acid footprint. And of those 21 amino acids, only one of them is, most of them are, I would say, 17 out of the 21 are absolutely conserved. 4 of the 5 are conserved, such as leucine to isoleucine, and only one of them is actually a non-conserved amino acid change. So the structure bears out the molecular virology approach that is being taken at this time. And then finally, we can ask the question, what about the variation? We're obviously deeply interested in the potential for resistance. We continue to try and select for resistance. And one of the things we're looking at is where does the sequence space vary according to what's been published. And so far, as you can see in yellow, that is tending to fall outside of this footprint domain. I actually don't think I have time to delve a little bit too much into this, but just so you're aware, S309 was the antibody we isolated. We did add in a half-life extension mutation, which is the LS mutation, which some of you may be familiar with, is being used in HIV antibodies. That's to extend the half-life so that, for example, if you were to give an injection in a nursing home, that antibody would persist, hopefully for many months. And then we've also made a second version, which we are planning to bring into the clinic, called 7832. This drug candidate has what we call the vaccinal mutation. I think many of you are aware of the work of Jeff Ravage, who has shown that not only does the FAB matter, but the FC matters. And I want to end with one quick slide on recent work that he has submitted. And this is being used by permission of the Rockefeller Lab. He has shown that, for example, now this is an influenza model where he has added in certain FC modifications. And what you're seeing here on the y-axis is weight change. As you know, by -20, the mice need to be sacrificed. And on the x-axis is the serum IgG concentration. And what you're seeing here is a 5-fold increase in potency by increasing FC interactions. Specifically, if you deplete CD8 T cells in these mice, this advantage goes away, suggesting that this antibody is actually enhancing a CD8 T cell response. So I think those are all interesting things. They're all preclinical, but that's why we're pursuing at least 2 different variations of this molecule into the clinic. And obviously there are many other antibodies companies who are pursuing their own antibodies. So I want to conclude by obviously touching lightly on the moderate to severe hospitalized trials. Everyone is familiar with remdesivir and the recent data showing a decrease in the duration of hospitalization. There's also the IL-6 data, which was interesting and seeming to show a trend towards an improvement in respiratory status, if not in mortality. Obviously, we do not have any hard mortality data for these particular drug candidates. I think everyone recognizes also the challenge. When these patients are this sick, it may be that it is very challenging to reverse late-stage disease pathology. It may require combination therapy, and we still have quite a bit of a ways to go. And to that end, I actually wanted to give a shout out to Laura Esserman. She is planning on doing a I-SPY COVID-19 trial to look at combinations of drugs in the network that she set up. So with that, I actually want to save a bunch of time for questions and really conclude by saying thank you once again. I realize it is unusual to have someone from industry present at an academic grand round, so I appreciate your indulgence, and thank you, and we'll take questions now.
Phil, that was really excellent, and it was extremely helpful. We are getting a few questions popping in, but I first want to turn over to Dr. Harrington, and he also, I believe, has a question for you.
Well, Phil, first off, thank you for joining us and welcome back to the farm. That we're always great to see that our alums are thriving. So welcome back. That was really a spectacular talk. And my question maybe gets more to science policy than the actual science itself. And I was struck by your comment that there are 76 vaccines in some sort of development and may not make it, most, if not if not all, won't make it anywhere. And I'm wondering, in the time of a national/global catastrophe, is there a mechanism amongst the vaccine making companies to come together and say, hey, let's pool our resources, let's pool our know-how, let's form a consortia, and let's put the bets down on the potential best winners? Because while the free market often results in great therapeutics, I really worry that we could go down a lot of rabbit holes here without gaining much success. So I have no idea, it's not my field, if there are such consortia that get put together, and I'd love your thoughts on that.
So let me answer that in a couple of ways. Obviously, in an ideal world, there would be more consortiums and more collaborations. I think that is something that actually is discussed at a high level between many biotech and pharmaceutical companies. I think the question is can you get everyone in the same to agree? I wanted to highlight actually a collaboration between Sanofi and GSK, who have been competitors in the vaccine field for ages, actually recently announced that they are working together on a COVID-19 vaccine. In some sense, that's an ideal marriage in my mind, because GSK has some of the best vaccine adjuvants on the planet. Their zoster vaccine is a good example of that, extreme immunogenicity even in the elderly, and Sanofi has made a lot of headway on the actual antigen, so being able to put those two together would be huge. But I guess it's like one of the problems is, of course, everyone believes their vaccine is somewhat better than the others, and the question is how do you adjudicate that? And imagine it, I'm actually imagining it from a regulatory perspective. From regulatory mandate, they need to look at every single vaccine. What position does that put them in when they have to spend 1 hour on the Janssen vaccine, 1 hour on the GSK vaccine, and 1 hour on a vaccine from a company they've never heard of? It's a very interesting policy problem right now. But of course, if you were to then institute a committee that would help them decide, you can imagine the kind of both political and economic pressure that committee would be under to understand why those decisions were made the way they're made. So I don't think we have a mechanism right now. What I am seeing is happening at the site level is that, for example, I was talking to Keith Flaherty at the Mass General and Dr. David Lai in Singapore. They are actually putting in committees within the institution to evaluate COVID protocols as they come through. Now, whether or not that's any better or any worse, I have no idea. But I think that is a reality that people will face as this deluge of clinical trials comes through. And then but I do think it is something that we've not had to worry about before. Now, everyone knows drug companies competing in the post-marketing space. My drug is better than your drug. What we're actually seeing happening is that competition is playing out before anyone has dosed a single patient. And that to me is just a mind-boggling situation.
Thank you.
Phil, thanks so much again. A few questions for you here. So here's one question, and paraphrase it a little bit. What is the expected timeline specifically for phase 1, phase 2 of vaccines? How long does it take a patient to have not to be diagnosed with COVID or for them to be declared immune successful from the vaccine? I think it's just a bigger question. One question I want to ask you, the most difficult one, is the timeline. From your perspective, of course, your unique perspective, what type of timeline, you addressed it a little bit, but pushing a little more, do you think we're looking at here? I know it's very early still. It's a tough question to answer.
So let me for the vaccine space, let me just use probably the best examples would be Moderna and the quote unquote Jenner Institute Oxford AstraZeneca vaccine, both of which, and I can't remember the exact status, but pretty much we can say they're starting dosing either now or have started dosing or will start dosing imminently. Those, and phase 2, I mean, not phase 1, but phase 2 in an actual efficacy trial. Those trials will really be dependent on the number of patients that they need to enroll. So imagine if you have a 8,000 patient study, it'll enroll a lot faster than a 30,000 patient study, but the likelihood that the 8,000 patient study may be underpowered is obviously higher. But I think most companies believe they can enroll that probably in a matter of months, and then they will probably be looking for, the quote unquote good thing is that you'll know whether or not it's efficacious relatively soon, because most people who develop COVID are, this isn't a progressive disease that takes 10 years to diagnose, right? It's the event rate is the clinical endpoint is rather easy to capture, whether or not it's symptomatic disease, these sort of things. So I think many of these companies are projecting they'll be able to have readouts by the end of this year. Whether those readouts are positive or negative, obviously, is unknown. And then the question becomes, when they read out, what kind of scale of availability will those vaccines have? I think monoclonal antibodies are potentially on a quicker time frame just because of the nature of the studies. But honestly, I think it's hard to say. And again, I want to emphasize that even if we, one of my fears, frankly, having the title of chief medical officer, is we're going to get to the end, we might actually have a monoclonal antibody that works. And then, the pressure to distribute, I mean, I'm not surprised, for example, I have no insights into why Gilead made their decision to donate all their supply, but I can imagine the pressure they would have been under to say, okay, well, why did you give it to this country, that country? Why did you give it to this hospital, not that hospital? There will not be enough, no matter how much we manufacture. And so to me, the concern would be, even after we show something works, let's say in the monoclonal antibody space in the fall or the winter, in the vaccine space in the winter, what's the next step? And that's what companies are probably spending the most time right now focused on, is to make sure that there's something available for compassionate use, something available for at least that initial period before you start ramping up. Because most of these vaccines and monoclonals, pretty much you need to start manufacturing now at scale, at commercial scale, in order to have that material available by the end of the year, which means you're going to be spending hundreds of millions of dollars on a drug and manufacturing, on a drug candidate and manufacturing it even before you've even sent it into your first patient. So that's part of the challenge right now.
Okay, excellent. It was telling that you talked about, even if we get to 1 billion doses, we're still going to miss a 6/7 of the world's population, and yet most of the companies right now, the goal, if they even, would be ideal to get to 1 million doses by the end of this year, and that's assuming all sorts of things that have to happen. So it's a very telling point you brought up. Can I ask another question, Phil? I had this was from Kyle Lowe. He asks, maybe I missed this, but the structure that Phil, that you showed of the S309 epitope, does it overlap with the ACE2 receptor binding domain? It seemed that the S3, I'm sorry?
It does not. And that's actually one of the things I highlighted, which is predictable from the sequence space. So if you actually compare the ACE2 binding site between SARS and COVID-19, it is highly variable between those two, but they still manage to both bind to ACE2. Since our epitope is neutralizing both of these viruses, it's not surprising that it falls outside of the actual ACE2 binding site. It does obviously bind to the receptor binding domain, and our feeling right now is that it's causing the receptor binding domain to achieve a certain conformation, which then prevents viral entry.
Gotcha. How is your antibody to be dosed? How often would one need to take it? I don't know. It may be early for you to answer that question.
So in the inpatient and the early treatment space, likely it will be intravenous. And the reason why I say that is because you don't. The last thing, in this many risks that you're going to take, the last thing you want to risk is that the drug doesn't get there in time. With a subcutaneous or intramuscular injection, it takes 7 to 14 days for the antibody to peak in your concentration. And that's obviously something you do not want to be dealing with in this type of setting. So definitely in the inpatient setting, it'll be IV. Likely in the early treatment setting, it'll be IV. When the prophylactic space, intramuscular may make sense. I think, from a distribution, again, this is now juggling competing priorities. From a use perspective, you would want it to be intramuscular, so you can simply give an injection and move on to the next patient. However, again, let's imagine you're an index patient in a nursing home where maybe 30% of the people are going to end up becoming infected. We don't know how quickly they're going to become, that transmission is going to occur. If you use an IM injection, and again, it takes you 7 to 10 days to reach peak, then maybe you'll either have to use more antibody than you necessarily think you would need to use. But of course, every doubling you do in the antibody concentration means at the back end, you have half as many doses you can distribute worldwide. So these are things that are quite challenging. And the answer is, we will probably go with a combination of IV and IM, depending on where the patient is and how much time we think they have. but I think also conservatively we'll end up with probably a higher dose than we actually, that may be absolutely biologically necessary, simply because, this is not a time also to be doing, infinitesimally small dose ranging studies. But again, we are mindful that that'll cut the supply in the back end potentially significantly.
Gotcha. Dr. Nick Leeper asks about intentionally exposing patients to to further accelerate the development of these vaccines. Is that something you can comment on?
Sure. By the way, I got a.
You got a heart.
Are you hearing me fine?
Yeah.
Okay. Sorry, I got a note that said my microphone might be not working. So that is a. Let me try and address that from an ethical perspective, and then address it from a pragmatic perspective. I think from an ethical perspective, there was actually a recent document, and I can't remember whether it was the WHO or a different organization that put out thoughts on what we would call a human challenge study. I think that there is, in a setting where we do not have any rescue medication that we know impacts mortality, it is going to be very challenging that if you were to infect 100 people and one of them were to die, remdesivir showed a mortality trend, but not a statistically significant one. As you know, the study was stopped early. It would be challenging, I think, ethically to deliberately infect someone knowing that there was not necessarily a medication on the back end. Now, of course, you could always argue, well, 99% of them won't die or 90% of them won't die, but frankly, I would not be I would personally not want to be that particular physician. But I think there's also a biological issue, which is that most likely no one is going to allow you to do this with a standard strain of COVID-19. It would have to be an attenuated strain of COVID-19. And there is so much data on the flu challenge models that pretty much they are not predictive of clinical efficacy, because once you attenuate the strain, it doesn't behave by definition the way that a normal strain would. In the case of flu, it's been attenuated to deliberately not infect the lower respiratory tract. To have a COVID strain that only impacts the upper respiratory tract would be, frankly, to challenge them with the common cold, if you would, which I don't think is what we're facing. So I'm not optimistic about that. Certainly, I would love it if there was innovation in that space, but I'm not counting on that to accelerate things in the short term.
Gotcha. Perhaps one or two more questions. Dr. Ron Lee asks, why are monoclonal antibodies not typically used as therapies for other viruses if they have advantages? Maybe that's just some of the unique situation we're in currently, or?
No, that's a great question. So antibodies are typically used, obviously antibodies work for prophylaxis. Whether or not they work for treatment, honestly, the first and, to my knowledge, one of the best examples is the most recent Ebola example, where it was used in moderate to severe patients and was able to prevent death. I think that we need to be careful as to what space we're talking about, in the sense that, for example, many flu antibodies have been tried in flu treatment and they have not succeeded, and I'm mindful of that. I would say, however, that flu has a different temporal course than COVID-19. It's often much more acute, much more aggressive, and then fades away quicker. This has a much more longer prodrome syndrome, but I think that is definitely a risk, and that's why I would say that earlier the better is always going to have a higher probability of success. But I think that there is reason to believe that it will work in treatment, in the treatment setting. I think that frankly, this has been a neglected space. It's one of the reasons why VIR was founded, is to be able to really explore the use of immunology in the setting of infectious disease. And that's exactly what we're doing.
Phil, thanks so much. As we hit 9 o'clock, I think we'll end here. I just wanted to thank you again for an extremely informative and enlightening talk about this. And we really appreciate taking the time to join us. And we hope you'll keep us updated on how things are going with you and any other perspective you have down the line. I also want to thank our panelists again for answering, once again, multiple questions. We'll get that sent out to you all. Again, I just wanted to put a plug. So Phil, thanks so much for really talking about vaccines and the work that you guys are doing in your perspective. It's been the #1 theme that we've had in the past weeks about vaccine development. We have another talk with vaccines on June 3rd from Kari Nadeau, working with the FDA as well, and so we'll come and talk more about this topic in the upcoming weeks. Again, Phil, thank you for the great, I didn't mention, but we had as many comments as questions of what a great presentation this was. And so it was really helpful. So thanks again to everybody. I hope you all have a great week, and we'll see you again soon.
Thank you.
Thank you. Bye-bye.